It's my pleasure to introduce our speaker of today. So today joins us Toman Borteçen. So he's a PhD student in the German Center for Cancer Research. His research is basically focused on the regulatory mechanisms behind the mRNA translation in cancer cells. And what we're going to be hearing today is about their work on basically creating or an semi-automated workflow to detect protein synthesis using a mass spectrometry. So with that, I'm very much looking forward to the talk, and I leave you with our speaker of today.
Yeah, hello everyone. First of all, I would like to thank, of course, the organizers for giving me the opportunity to present some of the work from my ongoing PhD project. So the presentation has this rather lengthy title, Semi-Automated Workflow for the Quantitative Analysis of the Newly Synthesized Proteome. And before we go into all the details of how this workflow came about, I think since this is a bit of a niche application and methodology, I would like to give a brief introduction into what exactly this tries to streamline and what my research is focused on. And for this purpose, I try to ask and answer a few basic questions here. The first one is, of course, why should you be interested in analyzing the newly synthesized proteome or protein synthesis in general? And of course, in many basic research questions, we work, for example, with cultured cells. And if we treat these cells with, for example, external stimuli or drug treatments, etc., or they experience stress, they will try to adapt to these changing conditions by essentially remodeling their proteome. And through the rechanged or remodeled proteome, they eventually obtain a different phenotype, which is adapted to the changed conditions. Usually, however, this remodeling of the proteome can take quite some time, meaning between several hours or even days. And if we are interested in our research in these initial responses of the cells, the analysis of the newly synthesized proteomes can give us kind of a snapshot of this initial proteome remodeling. And this can be quite valuable to understand and also distinguish the primary responses of the cells versus secondary responses which occur later on. So that is why it's quite a valuable technique. Um, in order to, okay, if we then want to carry out such an analysis of the newly synthesized proteins, or NSP for short, and we want to do so on a proteome-wide scale, there's a number of methodologies that are available. But essentially all of them rely on this concept of metabolic labeling. Metabolic labeling allows us then to essentially distinguish the previously synthesized proteins from the NSPs. Um, and as I mentioned, there are different methodologies that use this concept, but I will present here a little bit about the one that was developed and established in our lab quite a few years ago already. In this case, the method relies on a combined metabolic labeling strategy, which first of all uses stable isotope containing amino acids, or SILAC for short, which are incorporated into the newly synthesized proteins and therefore can be later through LCMS analysis be distinguished through the high resolution mass analyzer. Um, in addition to that, we also combine it with labeling using methionine analogs. The most prominent of these is azidohomoalanine, or AHA for short. Um, this non-canonical amino acid is also incorporated into newly synthesized proteins and allows us to perform a click chemistry-based enrichment of the newly synthesized proteins. When we perform an experiment and use this metabolic labeling, it is a very basic and streamlined procedure where initially you would culture your cells in normal medium, but then for the treatment or whatever process you're interested in studying, you will change the medium and perform a pulse labeling with medium containing this methionine analog that I mentioned, AHA. or heavy or intermediate isotope-coded amino acids, which get incorporated into the newly synthesized proteins. And then we can prepare samples for LC-MS analysis and study the newly synthesized proteins using relative quantification also. I mentioned now already that we can perform enrichments of these newly synthesized proteins. And you might also wonder, what's the benefit of this? In general, if we perform a relatively short time course experiment or perform the treatment of our cells in relatively short time spans to study these initial responses, most of the time we will have a situation similar to depicted here in the scheme. The overall abundance of the proteins between the two different conditions is most likely going to be very similar. However, there might already be quite significant differences in the newly synthesized proteins coming from either condition. And if we have such pulse-labeled samples, here's a scheme of how the raw data would look for the respective peptide ions coming from the proteins that we would measure in the mass spectrometer. In the case, if we just perform a pulse labeling and do not enrich for these newly synthesized proteins, the majority of our signal is going to come from these previously synthesized proteins, or rather their peptides, versus a small fraction of the signal we'll obtain from these newly synthesized proteins. Through this enrichment, we are able to essentially increase the signal of the newly synthesized proteins and effectively reduce the signal-to-noise ratio of the analyte we're interested in, which in this case is the peptides coming from the NSPs. As I mentioned before, the distinction between the different conditions here is not really a problem due to the use of high-resolution mass spectrometry. However, the main benefit here is definitely this increased signal-to-noise, which, as I'll show you, has a major benefit. So here's now some data in which I'm comparing essentially samples that I obtained from cells that were labeled for 4 hours, either without the enrichment of newly synthesized proteins, is labeled input here, or NSP standing for enriched newly synthesized proteome sample. And here in this panel A, we can see that comparing the number of quantified protein groups between the two samples, we have a minor benefit for the samples that were enriched, but this is not really all that striking. And more importantly is that we have much improved quantitative accuracy and also precision as a result of this enrichment of newly synthesized proteins. And as I hinted to before, this is due to this improved signal-to-noise signal or ratio that we obtain. So much as a background for the basic methodology that has been around for some time. It's very powerful. However, there are serious limitations also, which we believe also explain why this technology has not so widely been adopted by researchers yet. In my opinion, there are 3 major problems. The first one is that in order to perform such an enrichment for the newly synthesized proteins, we require quite a lot of protein input. Usually, most approaches will use something upwards of 500 micrograms of protein, up to even the milligram range. And all of these enrichments, be they click chemistry enriched or biotin, streptavidin pull downs, they usually require manual sample preparation, which is rather tedious and also limits the throughput and how many samples can be processed at the same time. Also, there's another issue that usually arises with the analysis of these enriched samples, and that is that they conventionally make use of mass spectrometry acquisition methods that require either the use of offline fractionation or long LC gradients, which really extend the measurement time, which is needed to obtain deep or relatively deep proteome coverage, which additionally limits the throughput of such analyses. I will talk about this last point towards the end of the presentation, but first I would like to explain how we try to improve upon these first 2 limitations in the sample preparation. So, as I mentioned, due to the high protein input requirements and also the limited throughput through manual processing, we sought to essentially improve the enrichment strategy by finding a more suitable bead type. And our end goal was essentially to scale down the dimensions of this enrichment procedure in the sample prep and also potentially to automate it. Conventionally, we use for these click chemistry-based enrichments cross-linked agarose beads or resin, which has a really high density of functional groups or alkyne groups in this case. Usually, per milliliter of resin, it will have something between 10 or 20 micromolar of alkyne available. These beads work fantastic, but of course, they require manual preparation or centrifugation steps are included in most protocols. There are some commercial alternatives, such as these magnetic beads, which are a layered polymer and magnetite particle, which of course on the first glimpse would seem to solve this problem, that we now have a magnetic bead type that can be used to automate and uh, essentially improve this enrichment. However, most of these commercial bead types are not really suitable for the analysis, since they have only a small number or small density of functional groups, usually in the nanomolar range per milligram of bead material. And as we found out the hard way, the binding capacity of the beads is really the crucial factor for obtaining high quality samples in the course of the enrichment. Because of course we are limited by how many of these AHA containing newly synthesized proteins we can covalently bind to the beads. So we really need, in order to obtain our goal and streamline and optimize this enrichment procedure, we need to obtain a high capacity magnetic bead, which allows us to make these improvements. So we were quite lucky to then find out that we can use these epoxy activated magnetic agarose beads, which we modify or couple with propagylamine to produce in-house relatively easily these magnetic alkyne functionalized beads. And these can be used just the same as we would the non-magnetic agarose beads for using click chemistry to couple the AHA containing newly synthesized proteins covalently to the beads. So using these magnetic agarose beads, it is possible to perform essentially the whole enrichment procedure in the scale of a PCR tube with lower amounts of material, but also crucially, through the use of a magnet, it is much easier to adapt this enrichment to a robotic liquid handling platform, which is what we did. So then, testing out this semi-automated enrichment protocol that we developed, it is possible to process up to 96 samples in parallel, and all of this in a PCR tube format. What we also wanted to show here is some data, is that instead of using upwards of 500 micrograms of protein material, it is possible to generate high quality samples with around 25 micrograms of protein input. A maximum threshold of protein identifications, for example, is already reached at 100 micrograms of protein material, which is still significantly lower than what is conventionally used. Furthermore, I also show some interesting data here in panel B, which is we like to assess the efficiency of our enrichments of the newly synthesized proteins by looking at the ratios of labeled or stable isotope containing peptides and their intensity versus the previously synthesized or background binding proteins, which do not contain the isotope labels. And what we can see is that even with very low amounts of protein input, we get the majority of our signal actually from the labeled peptides originating from the newly synthesized proteins, indicating to us that this protocol is very efficient in performing the enrichment of the NSPs, even in this lower range. Great. So now coming to the final part, where, as I alluded to, we also wanted to do some optimizations on the acquisition side. We had this problem that if we are capable of producing so many samples through the semi-automated sample prep, we will, of course, in the end have the bottleneck of LCMS measurement time. And using the conventional data-dependent acquisition scheme, which is usually used for these SciLec multiplex samples, this would require offline fractionation or very long gradients, which would lead to problems in the end because of long measurement times. There's one solution which we became attracted to, and this is the newly developed methodology and software tool for multiplex data-independent acquisition. There was a very nice paper which I encourage everyone to read, where they use this for non-isobarically labeled protein samples, and uh, we adapted this now to also be compatible with SciLec labeled samples, and carried out an extensive benchmark to see if this new methodology and software is beneficial for the types of samples that we produce. Um, I can only show a little bit of the benchmark here, not to make things too busy. But essentially, we found that this actually is very beneficial for improving not only the number of quantified proteins in our enriched samples, but also to still obtain really high quantitative accuracy and precision compared to the conventional acquisition scheme. So it is definitely highly beneficial for the types of samples that we produce. And taking together all these optimizations, being the preparation of the magnetic alkyne agarose beads, the metabolic labeling, plus some other processing steps, including the semi-automated click chemistry-based enrichment of the newly synthesized proteins, and the improved LC-MS measurement through Plex DIA. This comes together into this workflow for the quantitative newly synthesized protein analysis, which we also have given this this acronym name QuantPa for. You can read about it in the preprint that was also linked to this presentation, and we hope to have the revised version published very soon, hopefully. Finally, I would like to show one more piece of data where, now having established this optimized workflow, we applied it for the study of essentially a well-studied system to further add validity to the kind of data that we produce. For this, we treated HeLa cells with interferon gamma, a signaling protein from the immune system, and we measured the changes in the newly synthesized proteome at 5 different time points. What we were able to find in this data was that even as early as 2 hours after stimulation of cells, we're able to measure significantly upregulated proteins which are well characterized to be targets of interferon gamma. And through the number of different time points that we measured and processed rather easily through the automated procedure, we were able to show essentially that using this methodology, you are able to generate high quality data that provides valuable biological insights, especially, for example, for studying protein signaling. So, and with this, I would already summarize that in general, the enrichment of newly synthesized proteins, or NSPs, is a really valuable methodology that we believe is not used as much as it should be. It can be used to really get this snapshot of the initial proteome remodeling, even with short time scales, and it does so with improved quantitative accuracy and precision compared to the conventional pulse labeling. In our work and the QuantPa workflow that we developed, we were able to improve on some of the limitations of pre-existing protocols or workflows, being that we can now process up to 96 samples in a semi-automated workflow or manner, and we can do so even with relatively low protein input requirements, which really streamlines the preparation of these samples. Furthermore, by adapting Plex DIA to be compatible with SciLec labeled samples, we were able to improve the throughput and proteomic depth of the measurement of these samples. And we do so and acquire this data at high quantitative accuracy and precision. And I would also note to that actually that all the data that I've shown, and including this benchmark of the Plex DI technology, that all the data was acquired with a now 7-year-old Q-Exactive HF. So with a later generation or more modern mass spectrometer, you will get really impressive numbers, most likely. Okay, and with this, I'm at the end, and I would like to thank you for your attention. And I would also express my thanks to all former and current members of our group in the German Cancer Research Center, especially Dr. Thorsten Müller, who helped me with the automation of the sample preparation, and of course, Professor Jeroen Krijgsveld for his supervision and allowing me to work on this project. I'm happy to answer some of your questions or any that I can, but you can also, of course, reach out by email if you want to discuss or have any thoughts. And yeah, thank you.
Yeah, so thank you very much for your talk. It was super interesting. And before I forget, if you actually didn't catch the link to the preprint during the presentation, you can click on the banner up there. It's still available for you to go directly to it. So yeah, I would like to start then the Q&A question, and maybe I ask the first question.
Sure.
Do you have any idea of how many proteins you could be missing because the methionines are not exposed, or they are not, or they are losing their initiator methionine, or something like that?
Yeah, that's a great question. So there's for sure going to be some proteins that we won't be able to enrich through this click chemistry-based approach. It should, however, be in the low percentage range. If we compare the results from these enriched NSP samples compared to a standard proteome sample, the overlap in identifications is very similar. So generally, I would assume that the bias is not that strong. But it's something to be aware of course. If you're interested in particular protein targets, this is maybe something to be cautious with.
Right. Right. So then, Bernd Lan asks, how do you incorporate this non-canonical amino acid into proteins?
So when we work with mammalian cells, be it human, mouse, etc., this is essentially a brute force labeling approach. So we need to before we switch this media, as I explained, we need to have a short period in which these cells are in deprivation of the internal amino acid stocks. So we keep them for usually 30 minutes to 1 hour, depending on the cell type, in a medium that does not contain methionine, for example. AHA is incorporated into proteins through the same machinery as methionine. However, it is done less efficiently. Therefore, methionine depletion is crucial when labeling with AHA. But in general, the basic machinery of your cells is capable of doing it. I hope this answers the question more or less.
Cool. Then, so Hailey Sebaga asks, can you explain why the MACBIT approach needs way less protein than the original agarose approach?
Yeah, that's a good question. So in principle, between the binding capacity of the conventional non-magnetic agarose or these MACBITs, there shouldn't be such a big difference. However, if you're working with the non-magnetic version, you're usually limited to a larger Eppendorf tube, for example, where you have these centrifugation steps. And therefore, often a relatively large amount of reaction mixture is needed, and also protein lysate in general. The main bottleneck in the end is then how you can how many of these AHA-containing NSPs you can covalently bind to your beads. And in general, this is done very efficiently for both of them. However, just to the scaled down dimensions in the PCR tube versus in a 2 milliliter Eppendorf tube, for example, it becomes just more convenient and practically doable to work with lower protein amounts. I hope this answers the question approximately.
Yep.
But yeah, it's a handling issue.
Right. So now we have a question from Mila Lang.
Mhm.
In the test assay you made with interferon A, what was the stimulus and how big were your cultures?
Right. Okay. In this case, we were using interferon gamma um, as stimulus for we used HeLa cells, and all of them were grown in 6-well plates. So samples were essentially generated from about 1 million cells per condition. Um, okay.
So Xiao Yu Liu wants to know, method is this method suitable for other non-canonical amino acid incorporated proteins?
Yeah, I like this question a lot. Thanks for asking. So currently, of course, this is all optimized for the use of AHA or azidohomoalanine. Um, this is what we primarily work with in our lab, and it works very well for many of these cell lines that we use in cancer research. However, there are other possibilities to use, for example, HBG. But for this to be applicable, or also other applications with O-propaguropuromycin, for example, which you could also use to label these newly synthesized proteins, um, you would have to adapt essentially which types of magnetic agarose beads you're using. We are now preparing here alkyne containing magnetic agarose beads, but you could also imagine modifying the coupling procedure a little bit to then produce, for example, azide magnetized agarose. And this you could use, for example, of the enrichment of HBG containing newly synthesized proteins, or also this really promising looking new candidate beta ethanolserine, which seems to be really interesting as well. And this is something we are considering to try out in the near future. So thanks for the question.
Cool. So another question from Bernd, and is your method also applicable to other cell types?
Yeah, so in principle, you can use this for most cultured mammalian cells. It does depend a little bit. It works better with fast-growing or fast metabolizing cells, but in general, this should be working well with any mammalian cell type that we have tested so far. For prokaryotes, I cannot really say. I have never tried. I think there are approaches which also work with this. I would not be so sure about plant cells in the end because there's a lot of crazy biochemistry also with alkynes. Therefore, there might be some interferences. But in general, for mammalian cells, human, mouse, etc., no problem. It should work relatively well.
So Haley Sabaga has another question. Could you explain the Plex DIA approach for the mass spectrometry measurements? Why is it so much
Mhm.
faster?
Okay, so yeah, thanks for the question. Here I have to take a step back. I was, of course, jumping a little bit over many of the details of the mass spectrometry analysis and acquisition methods. Maybe if I want to give a short answer, I mentioned first what is conventionally done, data-dependent acquisition. This means that when we acquire spectra of the intact peptide ions, we then subsequently pick um, and use the quadrupole mass filter in our instruments to select and isolate essentially the peptide ions with the highest intensity. And it goes step by step. Individual peptide precursor ions are isolated and then through collisions with nitrogen fragmented. And these fragment ions are essentially used to sequence or match which peptide we have just analyzed. In data independent acquisition, and this is then part of this Plex DIA, the isolation windows of the quadrupole are larger so that multiple peptides or precursor ions in parallel get fragmented through these collisions. We acquire these more complex fragment spectra, and this is then analyzed through highly sophisticated software tools. It cannot be so easily analyzed by hand as data-dependent acquisition spectra. The novelty of Plex DIA is essentially that we have now a multiplex sample, meaning that our fragment ions will also have these shifts of the different isotopic labels. This complicates the fragment spectra even further. And just recently, there are a number of software tools which have been capable of analyzing multiplexed samples such as SILAC. However, this new Plex DIA is really showing extremely promising results for these highly complex multiplexed fragment spectra with SILAC or MTRAC labels, etc. And about the speed, I have to say, in principle, the measurement time is just if you perform, for example, if you want to obtain high proteomic depth in your measurement, you will need to, through DDA, either use longer gradients from the LC separation up front, or you have to do offline fractionation and measure multiple fractions of your sample subsequently. You can obtain similar or possibly even higher proteomic depth through DIA, which is all done in one measurement. And by not having to analyze these multiple fractions or long gradients, that's how you increase the throughput. And that's why this is also so attractive here in our workflow. If you generate this many samples and you can all measure the samples in a single injection, that's really convenient and kind of a nice solution, let's say. Yeah,
Cool.
I hope this answers the question.
I think that did answer the question. And we have time maybe for one last question.
Okay.
Right. So Milo Lang asks, is there any benefit not using your optimized workflow? So do you have any drawbacks to your methods?
I would say, of course, we have shown here a lot of great benefits, and especially when you want to use this newly synthesized proteome analysis for a number of conditions or different time points you want to study in your signaling event is great. However, if you do everything in this conventional large-scaled enrichment that I pointed to before as like what some limitations are, if you are not limited by the number of material you can produce in an experiment, meaning your cells can just be grown in larger dishes, etc., and you don't want to compare so many conditions, you just want to make 1 or 2 or 3 samples that you want to analyze, it might not be, or it might be good to do it on the manual, like large scale procedure, because that way you will produce so much sample that you can even analyze through multiple injections. Versus if you perform everything in our optimized and downscaled sample prep, you will have enough sample for, I would say 2, maximum 3 injections of the sample. So it's usually good enough if you also do the Plex DIA approach. You will get really nice depth, which should be sufficient. But if you want to do some technical optimizations or just want to have a large amount of your sample, maybe it's better to do the manual preparation at first. Otherwise, it's a good question, but I can't think of any other reason not to do the automated procedure. Of course, if you don't have the robot, then it's not so good. But otherwise, it's very convenient, and I would advise giving it a try.
Cool. Yeah. Then I would like to thank you once again for joining us today. This was super interesting to hear.
Yeah, thanks for having me.
Sure.